Okay, so this first lab is really just kind of getting you guys ready for writing some of the short reports that you'll have to write, and then the longer lab report that you'll be responsible for. So this isn't exactly the most exciting of labs, but I want to make sure everyone's kind of on the same page as far as using Excel, knowing kind of some of the descriptive statistics, as well as like more formal statistics, statistical tests, how to make figures, how to enter and organize data, all that stuff. So some of this you might know, some of it you might not know. Again, I just want to make sure everyone's on the same page. So I'm going to go through this really brief overview of describing data and statistics, and then tell you about what you can work on in lab, and then what the assignment is. So it's going to be very self-directed. I'll be here to answer questions, and yeah, I'll get into that in a minute. This will take like 15 minutes. Yes. Okay. Oh, yeah, that's fine. Yeah, thanks. I didn't see anyone over there. I went to the upstairs lab in our classroom. No worries. Okay, so yeah, I'm going to get started on this. This won't take, it might take like 10 minutes to go through this, and then I'll tell you all what you'll be doing today. Okay, got multiple screens here. All right, first, what we're doing today, talking about descriptive statistics. This is kind of an awkward room. I can't really get up there, so I'll use the mouse to, direct you as to what I'm saying. Okay, so descriptive statistics are not like formal statistical tests. We're mostly talking about like the mean when I say descriptive statistics or standard deviation. So I'll talk about what those are. We'll talk about two different tests that you'll be using this semester. You won't only be using t-tests and regressions, but I'll talk about when they're used and how you use them, and you'll practice that for your assignment for this lab. So for a t-test in general, you're looking at two different samples and looking at the means. Are they different? Regression is looking for a relationship between continuous variables. So if you don't know what a continuous variable is versus categorical, I'm going to talk about that. That's going to help you determine what kind of test to use. How many people have done a t-test before? Okay, not everybody. Regression, linear regression. Okay. I just want to make sure I'm not telling everyone things that everybody already knows. What's the point in that? Okay, some of the basics. So some terminology that I'll be using. We're looking at samples. So a population would be, say, all of the oak trees in the state of Georgia. If you want to look at the size of oak trees or something, you obviously can't measure all of the oak trees. So you measure a sample of the oak trees, which is just a subset. So we are talking about samples. In ecology, we measure samples. A variable is some kind of characteristic that you're measuring. If we're talking about the size of oak trees, it might be the height, or DBH. And I think everyone knows what data are, but they're just essentially the numbers that you write down, more or less. And I will point out here that data, the word is plural, so data are, it's not data is. I know it's a little bit weird and it sounds weird until you get used to it, but the singular of data would be datum. Hey guys. No, it's fine. I went over looking for people and I didn't see anyone, so, but just have a seat anywhere. So we're dealing with data today, and then throughout ecology, throughout this lab, we're going to be measuring different variables of different samples. So you will see these terms again. You don't need to memorize the definitions, just kind of have an idea of what I'm talking about when I use these terms. Okay, so first, descriptive statistics. You've heard of these before, probably in like high school math, mean, median, mode, and then range, which is on the next slide. But these are just summary statistics that kind of sort of give you a picture of what the data are showing. So summary statistics that describe the center are going to be the mean, median, and mode. The mean is really all we're ever going to use in here. That's the average. Median is the very middle number. If you're looking at, say, this whole set of numbers, the one right in the middle is the median, and the mode is the most frequently encountered. So this has the highest frequency, as you can see here. Median and mode, I don't think I'll ever say again, but median, we might use at some point, use it sometimes instead of mean for different reasons, if you have like a lot of outliers or something, but we'll mostly be using mean or average, same thing. So that describes where the center of the data lands, the center lands. And the spread, this other part up here, is also really important. So you want to know the average, but you also want to know how variable your data points are. And that brings us to the next idea, the range and the standard deviation. So range is typically like the highest versus lowest. We won't really be using range. That's more used in basic arithmetic classes. We'll be using standard deviation a lot. It's a more complicated thing to calculate, but thankfully Excel calculates it for you. We're not going to do it by hand. We're not going to do any statistical tests by hand because no one ever really does that anymore. So if we're looking at a couple of samples to give you an idea of standard deviation and kind of why it might be important. If you look at these two samples, just numbers of whatever, I don't know, some plants you found. Sample 1, you see here, sample 2, you see there, they have the same mean. So if you add them all up and divide by 1, 2, 3, 4, 5, 6, 7, if you add up those numbers and divide by 7, the means come out to 4. So you would say these samples are basically equivalent if you're looking at the mean. However, which of these do you think has a higher standard deviation without me having defined it? Yeah, 2. Why would you say 2 has a higher standard deviation?
Wider range of numbers.
Exactly. Yeah, that's all it is. So you're looking at numbers from 1 all the way to 7. That's a lot of variability. So this is measuring the variability around the mean. Whoops. I meant to animate that and I forgot. Let me go back. Let's just move the box. There we go. I meant to animate it so the box disappeared. Okay, so you can see the difference in standard deviation, 0.81 versus 3. That's a pretty big difference. There we go. We're going to look at standard deviation a lot. It matters a lot in ecology. If you have a lot of samples, the mean is kind of, or the numbers aren't that far from the mean. You don't have a lot of variability. You can be a little bit more sure about your actual mean. If you have a lot of variability, it's really hard in a bigger sense to actually get a statistically significant difference between two samples. So more variability of the data just makes you a little bit less sure about what the actual mean is, if you could really calculate the mean of a population. Okay, so mean and standard deviation, we'll see those many, many times this semester. All right, now discrete versus continuous data. This is where it's important to understand these two different categories of data in order to figure out what kind of test you're eventually going to do. So continuous would be, if you have continuous data, you're taking measurements, it would be something, you would have numbers that have usually decimals. It would be, I don't know another good word for it, continuous. It's not like you have 1 or 2 or 3. You could have anything in between 1 and 2, right? So you can think of it, temperature is a good example of that. Categorical or discrete data is in categories. So here, if you're counting the number of cats, like I said, you can't have 1.6 cats, right? If you're actually counting real cats, you have 1 cat or you have 2 cats, unless they're partial cats, which would be weird. So categorical versus continuous. These are synonyms: categorical, discrete, numeric, continuous. I think I mostly use categorical and continuous. They make the most sense to me. So you'll probably hear me using those terms the most, but you can put the data in categories, or the numbers are continuous and you can't put them in discrete categories. That's really important to know the difference there, because that'll help you figure out how to make your figures and then what statistical test to use. So if we're going to graph discrete data or that categorical data, it might look something like this. So along the x-axis, you usually have categories. This is bleak data. Sorry, I should probably change the information I have here, but 10 most common causes of death. So along the bottom, you have different categories that you put them in, right? It's not like you have a continuous number line along the bottom. On the y-axis, you're always going to have some kind of number. For our purposes, you're going to have a continuous y-axis. The x-axis, if you think of it in figure form, in discrete data, is going to be in categories. So bar charts are excellent for discrete data. You'll be making plenty of bar graphs. You can make more complicated ones, which we'll do later in the semester, where you actually group different categories together. So you're looking at multiple variables here, right? If the y-axis is what we're actually measuring, that's the number of native species in these different types of habitats. So where there's garlic mustard present, common buckthorn or honeysuckle, those are all invasive species. If the exotic is present, the non-native is present, or if the non-native is not present, you can see a difference and you can compare those two side by side really clearly. So when you're making bar graphs or any kind of figure, you want to think about how it tells the story. You could separate these out into two different graphs. You could have a full graph of just the dark bars with the exotic and then a separate graph with just the gray bars. But it really helps show that comparison that there are more species, more native species in each of these communities where the exotic is not there yet, right? Okay, so there's many different ways to do bar graphs. We're going to start pretty simply. If we're looking at two continuous variables, so like I said, the y-axis is always, I think for our purposes, always going to be continuous. It's always going to be some number scale on the y-axis. If the x-axis is also continuous, so here you have ACT scores. You can't categorize them. They can be anything in between, I guess, 10 and 40 or so. So there you'd create a scatter plot. So all of these are individual data points, right? So somebody got, I don't know, like a 19 on their verbal and then a 14 on their quantitative, something like that. So you would plot each point, and then from there you can draw a trend line, which we'll do today. But a scatter plot is the way to go if you have two continuous variables. Questions so far? All right. So we know how to kind of get an image of the data. If you can figure out what kind of graph you need to make, it'll be pretty easy to figure out what kind of statistical test you'll need to do. So a t-test is going to compare quantitative variables from two groups. So if you have categories, with t-tests, you have a categorical X variable. So if you can make a bar graph, you're going to do a t-test for the most part, for our purposes, starting out. The y-axis, the y variable, the response variable, is going to be numeric, like I said. If you don't have some kind of numeric variable, you can't really do stats as easily. All right, so category on the X, number on the Y. If you have two numeric variables, like that graph I just showed you, the scatter plot, you have continuous and continuous, X and Y, then you have a regression. And for our purposes, we'll just stick to simple linear regression. It'll show the relationship between the two variables. All right. So first, a t-test. What a t-test does is evaluates whether the means of two samples are different from each other, two categorical samples. Are they significantly different? That's what we're going for. Does null hypothesis sound familiar? Okay, good. The null hypothesis is always that there's no difference. So if you're looking at, we'll go back to the size of oak trees. If you're looking at the size of oak trees in an open field versus a forest, the null hypothesis would be that you don't expect any difference. There would be no significant difference expected. Abigail, do you have a question?
No.
Okay. Okay, that's fine. I thought your hand was up. Okay, that's fine. So null, there's always a null and an alternate or alternative hypothesis. The null is that there's no difference in height or whatever of the trees. The alternative would be that there is a difference, obviously. So you get a couple of values that you want to report when you run a t-test. Again, I'm focusing on the stuff that I think is really important. Other professors may focus on other aspects depending on how into statistics they are. I just want the basics. I want you to understand whether there's a difference in two samples. So you'll get, when you run a t-test, and we'll do that today, you'll get a value of t, a t statistic. You compare that to something called the normal distribution. We're not going to do that. That's how, I mean, basically Excel does that for us. We're not going to do that by hand. If it's more different than 95% of that normal distribution, then the test is significant. This bullet point is basically just kind of describing in more detail than you probably need to know exactly what the t-test is showing you. It's easy to misuse statistics, so I try to at least kind of describe what's going on. The important number is your p-value. Your p-value, if it's less than 0.05, that means you have a significant difference. That means that there's less than 5% chance that what you got, whatever difference you see, is due to chance. So 95% of the data is pointing towards, yes, there is a difference in these two samples. And this may not make a whole lot of sense right now. Once you run a t-test or two, I think it will make more sense, but what you're going to report, and I think I have this on another slide, the t-value or the t-stat, and then the p-value. Excel will spit both of those out for you once you run the t-test on your data. If your p is less than 0.05, you get excited because you have a difference. If there's no difference, that's fine too, but it's less interesting, I guess. There we go. A significant result means that the populations are different. You're looking for difference. Questions about that? T-test is probably the most simple statistical test. When you report the t-test, so yeah, this is where I have exactly how you'd report it. So when you do write up results for any of your reports that you're going to be writing, you want to report the test statistic and the p-value in a specific way. I don't want you to write in sentence form, the t statistic or test stat is this and the p-value is this. I want it like this. So look at that to go back to this when you're like, how do I report the statistics? This is your t-value. You'll find that in the table that Excel spits out, 2.36. That doesn't really tell you anything. Back in the day when you used to look at a chart to find out whether it was significant, it did, but we still report it. And then your p-value, you'll say p equals whatever. If this is less than 0.05, you know there's a difference. So this example was using tomato plants. Tomato plants had a height of 81, cucumber plants 29.5. You run a t-test on all of the data you collected from there, and you get this result, means they're significantly, the tomato plants were significantly taller than cucumber plants. A lot of times people just say there is a significant difference in height between tomato and cucumber plants, but that's helpful, but you want to know which are taller, right? So you want to specifically state the tomato plants, their mean is 81. They are significantly taller. So be specific. Okay, and regression. So this is where we have two continuous variables. Oh, here we go. Yeah. a continuous X variable and a continuous Y variable. Everyone knows that the Y variable depends on the X, right? So the X is not something that will change based on the Y, it's the other way. So the independent and dependent variable difference there, if you don't really remember that, go back and kind of review it. The X variable is independent. The Y variable is the dependent variable. It changes in response to X. We still have a null hypothesis, but now we're not looking at a significant difference. So with regression, we're looking for a relationship. Does X predict Y? Is there a change in a positive direction, or is there a negative slope? Not a significant difference. So with a regression, if you say the means are significantly different, it means absolutely nothing, because that's not what you're looking for. You're looking for a relationship or no relationship. Again, P less than 0.05. You can run a regression. You look at all your data, plug it into Excel. It'll tell you what your p-value is. If it's less than 0.05, same thing. If it's less than 0.05, the test is significant and there is a significant relationship. So yeah, the bold there, significant results. If you get P less than 0.05, it means that X predicts Y. That just means that there is a relationship between X and Y. It's not totally random. So for example, a plant might grow taller if you add more fertilizer. So the X variable would be the amount of fertilizer you add. As that goes up, the plant height should also go up. That's a relationship. Questions so far? Reporting a regression, you don't have a t-stat, you have an r-squared. R-squared will tell you how well the data points, remember that scatter plot I showed you, how well they fit the relationship. And then you still have the p-value. Oh, so here we go. If you add nitrogen and see how that affects tomato plant height, the r-squared being 0.94 means that it's a 94% fit, basically. The data fit the relationship really well. P is 0.001, so that's significant. It's a significant relationship. If you add more nitrogen, you get bigger tomato plants. Okay, you're going to make tables and graphs today. They can be kind of a pain in Excel if you haven't done them before, if you run into issues. I'm here to answer questions. Some general information about how to make figures and tables. So graphs are typically called figures. I don't know why we don't call them graphs in papers, but they're just figures. They always have a caption. This seems really particular, but this is how it's done in all of the scientific literature. Captions for tables are written above them. So they describe what the table is showing. For a figure or a graph, you put it below the graph. I know it's really nitpicky, but I'll take off points if you do the opposite. So put your captions above tables and below graphs. So once you've made the table or the graph, you're not done. You have to write a caption. And I have some examples for you. You should be able to interpret the table or the figure without having to read the whole paper. That's the idea of the caption. So explain it as well as you can. Include units in the tables and the figures. If you're making a graph, you have to have titles for the axes. So the y-axis has to have a title, so you know what those numbers mean. The x-axis has to have a title. But this is important, the whole graph, if you have a graph, you don't want a title at the very top of it. That's sort of elementary. So you don't put a title at the very top of the graph. All of that information goes in the caption below it. So here we go. Here's some examples of tables and graphs. Caption above the table, it's telling you exactly what is being shown in the table. It's more detailed than you're probably going to originally want to put in a caption, but just explain what's being shown. I think tables are fairly straightforward. We don't have any, this is just a count, I think, or percent pond area. Yeah, so there's no units, but if you had units, if you were measuring like height, you'd have meters or meter squared. If you're looking at area in the column headings, here's the T-stat for all of them and the p-value. All right, table. Here's some complicated looking graphs. Don't worry so much about the complicated figure in there, but here is the caption, goes below the graph. A y-axis label, say what's on the x-axis also. We'll talk about, and actually you'll be making these error bars, which show the standard deviation. So if you've ever seen these on a bar graph and you're like, what the heck is that little bar above it or below it? Sometimes it'll show it below it as well. That shows you how much spread there is around the mean. So anytime you have a bar graph, this line here at the top of the bar, that's the mean, but you know there's some kind of spread in the data, right? There's a standard deviation. So that's what this is showing. Every time you have a bar graph, you have to have error bars. Make a note of that. It doesn't make any sense without error bars. This is a different kind of setup. I think it's percentage. So won't really be doing much percent stuff. Okay. I know that was probably the most exciting presentation you've ever seen. I would say use that and go back to it if you're like, how do I report, the t-test results? Or how do I, if I have two continuous variables, what kind of graph do I make again? Use that, that's on Canvas. Use it as a reference. Other stuff I have on Canvas, I haven't signed in, should look something like this. The handout, so this is where some self-direction comes in. If you guys want to do this today, you can stay here and work on this. This takes you through a lot of different information. If you don't know how to use Excel, you don't know how to calculate an average, you don't know how to run a t-test. This is really wordy, and I apologize, but I wanted to walk you guys through it step by step. You can work through that. That's the idea for lab today. That's what you should take your time doing. And it'll tell you how to calculate standard deviation to make the error bars. So work on that. You don't have to turn the results of this in. I can check and see if you're on the right track, if you're getting the right answers, but you can see the screenshots for what you kind of should be getting in here. If you don't know how to do any of this, definitely spend some time. If you don't know the chart elements menu, if you're not familiar with that, that's where a lot of your options for graphs are going to be. Definitely read through this and do what you think you need help with. Are you guys aware of the data analysis tool pack at all? Okay, that's how you're going to do your stats. So to run a t-test, to run a regression, to run an f-test, which we'll do, that I didn't talk about, we'll do that later in the semester. You need to install the data analysis tool pack. So that is on page 8. And it'll tell you how to do it. There's some kind of weird quirks with it, but you won't be able to do a t-test or any of the other stats tests unless you install that. Once you install it on the computer, you can use it. I don't know if you sign back in with your same name, if it stays on these, but if you have your own computer, it'll stay on there once you download it. So you'll run a t-test, you'll run a regression, you'll make a scatter plot, you'll put a trend line on the scatter plot along with the y, or the equation and the r squared. So there's a lot of pieces in here. This shows you what the output will be for the different statistical tests. So this is a regression. A regression is tricky. The p-value that's listed here is not the p-value that you want. So when you're doing a regression, this is not going to be your p-value. Okay, well, at least that one. So read through that. It has a description of everything that you need. You are going to make error bars. Error bars are such a pain. You can't just put the regular standard deviation on there. you have to make them custom. This has the description for how to do that. A reminder of the pieces for figures and tables that you need to put in there, and some examples. So that's the Lab 1 handout that's on Canvas. You don't have to turn the results of that in. That's for practice. If you don't know how to do some of that stuff, definitely practice it there and I can help you. There is an assignment that I put on Canvas as well. Basically 3 questions. You're going to be doing a lot of that same stuff. So this is what you're going to turn in, the Lab 1 assignment. You are going to make a bar graph using these data. Then you're going to make a more complicated bar graph. So the first bar graph, you do not need error bars because these are just counts. For the second bar graph, you do need error bars. That's going to be more complicated. And then the third question is a regression question, so slightly different. You can put those all, you can do them however you want. You can upload an Excel file. You can upload a Word document with your figures and your statistics, results, etc. However you want to, just upload it to Canvas. It'll be due before we meet for lab next week. So you have a whole week to do it. I think that's it. I put additional stats help if you need specifically statistics help on Canvas. I put another document on there to help you with t-tests and regressions. And I even circled what you need in each of those printouts. So that's a helpful additional thing that I'm not really going to go over. Yeah. So for statistical tests, that can be really helpful. Okay. So you can work on the handout, you can work on the assignment. If you're working on the assignment and get stuck, I'm going to say go to the handout and practice it there. But I can answer questions for you if you're getting stuck on stuff.